Good morning, everyone, or I guess maybe it's good afternoon for many of you. As Ishwar said, my name is Obi Griffith from WashU, and I'm going to give you kind of an update on where we are with CIViC and accelerating the expert crowdsourcing of cancer variant interpretation. So this is background I think everyone in the audience is really familiar with the advent of this massively parallel sequencing. We have now largely productionified or automated the rapid identification of massive numbers of somatic and germline variants in cancer. And for many of us, we find that the major bottleneck is now at this interpretation and report generation and clinical application stage, where the genome analyst and healthcare providers are kind of deluged with this massive amounts of genomic and other molecular information and trying to interpret it in a clinical context. Clinical interpretations of variants are being produced by various entities, but many of them are done still in private academic silos or restricted access commercial databases. And these interpretations have limited provenance and little mechanisms for feedback or community involvement. And so we tried to address this problem by creating an open public domain effort for cancer variant interpretation. And our solution to that was the CIViC resource. So CIViC is available at civicdb.org. The idea is that as variants are discovered and evidence of their potential clinical relevance is developed in the biomedical literature, we can curate that knowledge into structured evidence using the CIViC curation platform, where it goes through a community curation process of discussion and revision and ultimately rejected or accepted to be part of the knowledge record. And that allows us to produce gene, variant, and evidence summaries, which can be incorporated into various research or clinical workflows downstream. The ultimate goal is to provide, sorry, did I mess that up? Do you still see my slides properly?
Yes.
Okay, I was just trying to move something out of my way. The ultimate goal is to provide assertions of a variant's clinical significance. Right now, we support evidence of a number of types, including predictive of drug response, diagnostic, prognostic, predisposing, oncogenic, and functional. And then these assertions can be assembled together or synthesized into a kind of state of understanding that we call an assertion. Okay. How do I get back to my slides here? There. So I thought I'd give you a quick tour of the CIViC user interface. So this is a view of one of the main pages, the gene page. Along the left here, you can see navigation to the top-level concepts in the knowledge base, like assertions, evidence, variants, genes, and so on. You can also perform a quick search across all of these entities. You can access our copious help documentation. You can optionally log in using Google, Orchid, or other methods. And then if you wish, you can contribute. So you can add or submit new evidence or assertions. You can make comments. You can propose revisions. Here we're seeing an example of a gene summary. So this is a curated summary of the clinical relevance of this particular gene. We then pull in details from my gene info to kind of supplement that with standard gene information. And then we can see a list of the variants that at the time of the screenshot had been curated as potentially relevant for this specific gene, ERBB2 or HER2. So let's just look at an example of one of those variants for illustration purposes. This is a variant page. Again, we have a variant summary, which is a curated summary across the evidence for that variant of its potential clinical relevance. We have here, again, metadata that's pulled in from my variant info, allele registry, ClinVar, and other sources based on the manual curation of coordinates for that variant done by curators in our community. And then you see a list here of individual evidence items, these structured evidence items that pertain to that variant. Again, if we just drill down and look at a single example, here's a very simple evidence item, which is an in vitro study showing potential predictive to sensitivity or response of this variant for the drug neratinib in breast cancer. So this is one of many different types of evidence statements that has a sort of short, human readable, can be as little as a sentence or as much as a short paragraph. And then pretty much everything else is structured elements using published ontologies or structured vocabularies. The final level is the assertion. So at the assertion level, we take multiple lines of evidence and assemble them into a kind of state of the field understanding of the clinical relevance of that variant in a specific disease context. So like a specific subtype of cancer for a specific therapy. And at this level, we can link it to other guidelines like the AMP-ASCO-CAP guidelines, where we establish its level according to those guidelines. We also can link it to relevant NCCN guidelines, FDA approvals of companion diagnostics and drugs, and so forth. Finally, this is just showing you a snapshot of what it looks like to submit evidence. So this is using our fairly sophisticated curation platform where you can, this has been pre-entered, but you can assemble molecular profiles, you can choose an evidence type and a significance and select a drug. Most of this is done very rapidly with look-ahead searches. You can establish an evidence rating and create the evidence statement, add comments, and so forth. Once that is submitted, oh, actually, there's some additional functionality I want to point out. If you start doing some of this work and the system realizes that you're entering evidence that is highly similar to evidence already in the knowledge base, it will warn you about that, so that can save you some time. And I also want to show you this new functionality for the construction of complex molecular profiles. So not only does CIViC support sort of single variant interpretations, but also interpretations for arbitrarily complex combination of variants. So here we're looking at a simple example of a variant and another variant and another variant being interpreted in concert. So once you submit that evidence item, others, expert editors in the community would review that and they can reject or accept it, or they can propose changes to it. So they can submit revisions, and then the user or other editors can view those changes with a kind of diff view and decide element by element whether to accept or reject those revisions. And through that process, we get slowly towards an accepted evidence item or assertion. So that's an introduction to what CIViC is and what it looks like. But what really distinguishes CIViC, we feel, is its really strong commitment to an open sharing philosophy and a collaborative community approach. So CIViC assembles a community of researchers, clinicians, and others who work together through a public forum, through open discussion, using curation standards and expert review to establish, we hope, a community consensus. And this is all done completely transparently. So every contribution, every comment, every revision is all fully publicly visible with who said what and when. And then the knowledge is all highly accessible, both through the free and open web interface, but also through APIs, which serve up the structured data. Again, it's freely accessible. And importantly, it's made available with a public domain CC0 license. So that really means that anyone, commercial or academic, can use it for basically any purpose without even needing to ask us. And we really feel that this open model has been an advantage in terms of widespread adoption. So we've had a very broad integration into the ecosystem of cancer variant interpretation. We have a large community of contributors, hundreds of contributors from different groups and organizations. We'll talk more about ClinGen later. This is allowing us to generate, we hope, a useful knowledge base, which is then accessed clinically through multiple clinical utilities, commercial utilities, and data clients, both academic and commercial. Things like Cravat or CBioportal pull CIViC data in and display them natively. We benefit from the use of a number of published on guidelines, and we also extensively use appropriate ontologies and regularly contribute back to those ontologies. And these journals, one journal is already recommending CIViC for variant interpretation or dissemination of variant interpretations and publications, and we're working to try to get other journals on board with that concept. So to date, we have hundreds of contributors, thousands of web visitors, millions of API hits, and this group of contributors, of about 350 contributors, has built a knowledge base of almost 10,000 evidence items representing 500 genes and more than 3,500 variants. So just to give you a couple examples of CIVIC's integration with different tools, this is a view of protein paint.
What is this, Daddy?
Excuse me. Buddy Daddy's talking right now. Okay, can you go find Mom? Okay, thank you. Okay. Sorry about that. Okay, so this is what CIViC looks like in protein paint. So if you go to protein paint, there's a track. It's one of a few tracks that you can pull up, and you can see clinically relevant variants according to CIViC in line with other key data sets like COSMIC and ClinVar. And also we link out to protein paint to give you those views from every CIViC gene page. Another example of an integration in the community is with more of a clinical use case. So this is the personalized oncogenomic study at BC Cancer. So this is a strong collaboration we have with the group in Vancouver, who are leading, I think, one of the largest formal clinical trials utilizing whole genome and transcriptome sequencing to inform diagnosis and treatment in the advanced setting. And their genome analysts rely on CIViC evidence for interpretation of variants found in their patient tumors. And they also contribute knowledge to CIViC based on POG findings and research conducted during case analysis. And they've really been strong contributors back to CIViC. So this is a really great example of a sort of virtuous cycle where they pull in CIViC knowledge for their own interpretations. And then as they discover new things, publish case reports, or just find relevant literature, they submit it to CIViC. So again, CIViC is one of their largest sources of knowledge, and they've made many contributions. This is an example of a case study where they had a cholangiocarcinoma patient with an NRG1 fusion that had a knowledge base match to CIViC evidence supporting sensitivity to afatinib. And that afatinib treatment in the metastatic setting led to a strong and durable response. That led to a nice case report, which you can find. I think it's now published. I mentioned ClinGen Somatic. So we've partnered with ClinGen to really accelerate the recruitment and organizing of expert groups. So the goals of ClinGen Somatic are to standardize curation and interpretation through the development of a uniform implementation of guidelines, collaborating with groups of experts, so-called variant curation expert panels, and then enhancing the usability and dissemination and implementation of cancer somatic knowledge through integration and interaction with these other groups. This is an overview of what that looks like and organizationally. So we have the Somatic Cancer Clinical Domain Working Group led by Malachi Griffith and Ramaswamy Govindan, with Jason Saliba as a coordinator. And we get oversight from a Cancer Variant Interpretation Committee, the Somatic Executive Committee, and the ClinGen Oversight Committee overall. And they are helping us to develop, and we have developed, a 4-step process for establishing somatic cancer variant curation expert panels. So we define the scope and goals. We develop specific variant classification rules in a gene and disease specific manner. We pilot those rules and then implement them in working expert panels. And so far, these have been launched from 3 task forces covering pediatric, hematologic, and solid tumor, and focused so far on the genes and disease areas listed below. So that's been a great source of collaboration and expert knowledge, which ultimately submits evidence and variants and assertions to CIViC, and they also get deposited in ClinVar. That's been hugely productive. So the ClinGen somatic group has submitted, I guess, nearly 1,000 evidence items so far. So a substantial fraction of the knowledge base, and been really active also in revising and commenting and suggesting new sources of knowledge. We're working together with the FDA to develop a subset of that ClinGen curated content as an FDA-recognized genetic variant database. Right now, we're working to get at least one, hopefully several SCV steps through that fully approved 4-step process and a number of requested SOP docs so that we can resubmit and hopefully get that approval. I did want to just mention that we have done a complete overhaul of both the front end and back end of CIViC recently. What this means is that we have a new UI design, which is more flexible in terms of searching and hopefully has more intuitive browsing features, but also has a totally new backend API, a GraphQL-based API that allows for a more user-friendly exploration of the data model. This does come with the caveat, though, that as a totally new API and with significant changes to the data model, we have basically diverged from our old V1 API. We've been maintaining both for about a year, but all new content, new data is going and only accessible through the V2 API. So I highly encourage you to, if you're using CIViC data, to become familiar with the new API and convert your workflows to use that, because the old API will go dark in November. We have extensive training docs with the Read the Docs site. We have published SOPs, a growing collection of videos in YouTube, and very regular training sessions where we try and disseminate knowledge about how best to use CIViC and contribute. And we're also now developing a pilot course using the ITCR training network tools. So they have this really cool resource called Otter, or open source tools for training resources, where you can develop pedagogic materials in Markdown and then cross-publish to Bookdown, LeanPub, and Coursera. So we do have a pilot course at course.civicdb.org, which we're starting to flesh out. We organized multiple hackathon and curation jamborees, and I did want to just make an announcement that we have another one of those upcoming at the Cancer Genomics Consortium meeting in a few weeks. That's actually August 13th to 16th. And if you're interested in learning about that, you can find information at cancergenomics.org. There are a few spots available still for that. So what's next? We've seen this incredible growth in the use of CIViC and accessing CIViC, in contributions, in data clients integrating CIViC, the growth in evidence of the knowledge base itself, and our own outreach efforts. One thing that's been a particular challenge is the growth in contributions and content has somewhat outpaced our growth in trained editors. So we're really trying to focus on improving that situation. So we have new tools that we're developing focused on optimizing the editor-moderator experience, including a kind of new dashboard view that will allow them to delegate tasks to other editors or editors with different levels of abilities. We'll also have the ability to quickly review outstanding tasks or tasks that may be blocking other entities from being accepted or completed. And then we're also working, again, with our friends at the BC Cancer Agency in Vancouver to develop text mining and AI-based approaches to think about how we might begin to automate some parts of the curation process. And so the first step to that was to develop a data set where we kind of link the curated evidence back to the underlying supporting text in the publications. So up till now, we have not been directly quoting or storing that information. But through this really great tool called Hypothesis, we're now able to highlight individual sentences or even sections of tables or figures which support the evidence item that's being curated. And so now you can curate a paper, you can tag those sentences with the evidence item ID from CIViC, and there's a Chrome plugin that then allows you to view those supporting statements in line with the curated entities in the CIViC interface. And that's allowing us to develop an NLI data set that Kara and Stephen at the BC Cancer are building models that would ultimately hopefully allow us to perform some automated actions, like automatically rejecting an evidence item that is sort of predicted to be beyond rescuing, or automatically identifying problem elements with that. So that's a quick update on the CIViC resource. I just want to acknowledge my group, our collaborators, and especially the CIViC community of hundreds of contributors, and our funding from the NCI. And I'll be happy to take questions. Sorry, it's a
Thank
bit of a
you,
family here.
Thank you so much. There is one question online that I will raise to you, I think, which kind of ties down to, crediting community of contributors. So what is the incentive for researchers to become reviewers, and also contributions map back to contributors?
So great question. Contributions definitely map back to contributors. So that's all tracked and presented. So as you navigate through any part of the CIViC resource, you will see, who submitted that evidence item, who suggested revisions, who accepted it. We track contributions also through kind of user profiles and leaderboards. We are working to re-implement a badge system, which is currently not live because of the massive switchover from V1 to V2, but that's high on the priority list. In terms of incentivizing or recruiting new editors to perform that important role, we benefit a lot from collaborations like that with ClinGen Somatic, where it becomes quickly apparent that for them to work through the cycle of developing their rule sets and proceeding with curation, it's really valuable to have people within each of the expert panels get to that level of editor. So we have criteria for how you qualify as an editor. There's training that's required. There's certain kind of tests that have to be passed. So we try to curate at least 1 or 2 editors in each of those groups, and that sort of benefits the group, which I guess incentivizes them to nominate people to become those editors. But in terms of the wider community, yeah, we're really dependent on people just finding the resource useful enough. They typically start contributing, and then over time become such important contributors that we will nominate them for training to be promoted to an editor. Yeah, that, as I mentioned, is one of our key challenges. How do we recruit and retain editors for that important role?